Thanks, how are you guys? I hope you people are doing fine. Okay, so the last time we looked at some terms that can be used when it comes to carbohydrate metabolism. We looked at glycolysis. We looked at glycolysis, where we said glycolysis is the process of breaking down a glucose molecule into two pyruvate molecules while storing the energy that is released during this process as ATP. Okay, ATP and NADH, that's nicotinamide adenine dinucleotide. So we'll continue on the terms that are associated with carbohydrate metabolism. Today we are going to be looking at gluconeogenesis. Gluconeogenesis. So from the word gluconeogenesis, genesis, build up. So gluconeogenesis is a metabolic pathway that results in the generation of glucose from non-carbohydrate carbon substrates. So glucose is gotten from non-carbohydrate substrates in order for there to be energy for the body to do work. So it is a ubiquitous process present in plants and animals, fungi, bacteria, and other microorganisms. In vertebrates, gluconeogenesis occurs mainly in the liver. So it occurs mainly in the liver and to a lesser extent in the cortex of the kidney. So it's not just the liver alone, but more of gluconeogenesis is done in the liver, and a lesser extent of it is done in the cortex of the kidneys. It is one of two primary mechanisms, the other being degradation of glycogen. That one is known as glycogenolysis. So when you hear glycogenolysis, the degradation, breakdown of glycogen in the liver to give rise to glucose, while gluconeogenesis is building up of glucose from non-carbohydrate substrates. So let's continue. Now, the reason for this is to avoid low levels, which is hypoglycemia. Okay, in ruminants, because dietary carbohydrates tend to be metabolized by rumen organisms, gluconeogenesis occurs regardless of fasting. So there are some animals where gluconeogenesis occurs, not just because the person is fasting. No, when you are fasting, there is depletion of glucose, so the body is supposed to produce glucose from non-carbohydrate substrates, or maybe break down from the liver, which is glycogen, and be able to sustain the body until the person eats food. So in ruminants, because dietary carbohydrates tend to be metabolized by rumen organisms, gluconeogenesis occurs regardless of fasting, low carbohydrate diets, exercise, etc. In many other animals, the process occurs during periods of fasting, like in humans, starvation, low carbohydrate diets, or intense exercise. In humans, substrates for gluconeogenesis may come from any non-carbohydrate sources. So I've said before, any non-carbohydrate sources that can be converted to pyruvates. And intermediates of glycolysis. So in humans, substrates for gluconeogenesis may come from any non-carbohydrate sources that can be converted to pyruvate or intermediate of glycolysis, which we talked about at first. So for the breakdown of proteins, these substrates include glucogenic amino acids from breakdown of lipids, such as triglycerides. They include glycerol, all-chain fatty acids, and from other parts of metabolism, which includes lactate from the Cori cycle. Under conditions of prolonged fasting, acetone derived from ketone bodies can also serve as a substrate, providing a pathway for fatty acids to glucose. Although most gluconeogenesis occurs in the liver, I've said it before, most of the gluconeogenesis occurs in the liver. The relative contribution of gluconeogenesis by the kidney, the cortex aspect of the kidney, is increased in diabetes and prolonged fasting. So we said before that most of the gluconeogenesis occurs in the liver and a little extent from the kidney, the cortex of the kidney. But in some conditions, like in diabetes, in diabetes and prolonged fasting, that the kidney can contribute even more, and that will be dangerous for the body. So the gluconeogenesis pathway is highly endigonic until it is coupled to the hydrolysis of ATP or guanosine triphosphate, effectively making the process exogenic. For example, the pathway leading from pyruvate to glucose 6-phosphate requires 4 molecules of ATP and 2 molecules of GTP to proceed spontaneously. These ATPs are supplied from fatty acid catabolism via beta oxidation. So in a nutshell, all we have been talking about since is gluconeogenesis, which we said getting of production of a glucose from non-carbohydrate processes in order to avoid or to prevent hypoglycemia, which is low glucose level. So the next one we're going to be looking at quickly is glycogenolysis. I've mentioned it before. No, I mentioned glycogenolysis before, so we'll just look at it briefly. It's the breakdown of glycogen from the glycogen store in the liver. Now, in the liver, muscles, and the kidney, this process occurs to provide glucose when necessary. A single glucose molecule is cleaved from a branch of glycogen, and it is transformed into glucose 1-phosphate during this process. This molecule can then be converted to glucose 6-phosphate, an intermediate in